Hello everyone, I'm Xiong from Chemical Engineering Department, and today I'm going to share some of my research results looking into the effects of N-glycosylation and on protein properties and molecular dynamics of a plant-made anthrax decoy protein. So first thing is, I want to explain why we use plant for protein production. There were several reasons for that. First, plant is intrinsically biosafe, as there is no chance for mammalian cell pathogen to infect plant cell. It is also capable for making post-translational modification, which cannot achieve using bacterial fermentation. It is also quite inexpensive compared to cell culture system, which often requires sterile condition and complex bioreactors. It is also linear scalable. By simply adding more plants to a greenhouse, you are getting more production capacity. And the most common way of using plant for protein production is through transient expression of protein. This method is realized by agrobacterial infiltration. First, we engineer the agrobacteria to carry gene of interest. Then we infiltrate our plants with the engineered agrobacteria. Then the gene gets transferred into plant cell and will express at really high level within a week. Using this method allowed us to shorten the development time, which allowed us to go from the gene construct to a purified product within a month. The picture on the right is showing the plant growth chamber from iBio, and those plant is used for protein production. Because the protein of interest is a protein against the anthrax, so what is anthrax? Anthrax is a severe infectious disease caused by anthracis, Bacillus anthracis. Anthrax spore can be produced easily and used as a biological weapon agent, which has over 50% of death rate even after aggressive treatments with antibiotics. So anthrax secretes anthrax toxin, which has three components, including protective antigen, edema factor, and lethal factor. The cytotoxicity process starts with the binding between human cell receptor of anthrax, or CMG2, to protective antigen. Once those two protein binds, the protective antigen gets cleaved, leaving a 63 kilodalton fragment attached to CMG2. And then this protein complex will self-assemble into a prepore, allowing the binding of lethal factor and edema factor. And once those two bind to the protein complex, they will get into the cell through endocytosis to cause cell death and edema. The way the decoy protein works is by making the binding relevant region of CMG2 recombinantly, and which can neutralize PA and preventing toxins from endocytosis. So on the left is a schematic representation of our decoy protein. We make this protein in Fc fusion form, which composed of the binding relevant region of CMG2, a flexible linker, and Fc of human IgG1. We make it an Fc fusion to simplify the downstream processing as well as giving the molecule a longer circulation half-life. By targeting the protein to different subcellular locations, we're able to produce three types of N-glycol form of the same protein. The first protein is targeted to apoplast, resulting in plant complex type N-glycol form. The second one is targeted to ER, resulting in oligomannose type N-glycosylation. The third one was by making a point mutation on the N-glycosylation site, making the protein aglycosylated. Once those genes construct into the same agrobacteria, they will express under identical condition, and the expression level is shown to the right. As you can see, the APO variants and the ER variants has a higher expression level compared to the A-gly variants, and we think that's mostly likely because N-glycosylation protects the protein from proteases cleavage by covering those protease sensitivity sites. And when you remove the N-glycosylation, those sites are more accessible to proteases, leading to a higher chance of degradation and a lower accumulation level. Then, to validate the function of our protein, I perform a toxin neutralization assay to find the effective concentration of all three proteins against the anthrax toxin. As shown to the right, the ER variant has a higher EC50 compared to A-gly and APO variants. And we have three hypotheses looking into this phenomenon. The first thing is Fc gamma receptor may be contributing to the toxin neutralization process by interacting with the Fc domain, and this interaction can be glycosylation dependent. In order to examine this possibility, we did a toxin neutralization assay with the Fc receptors blocked. If there's a change in the EC50, meaning that Fc and Fc gamma receptor is playing a role in this toxin neutralization assay. However, as shown on the right, the control curve compared with the curve with Fc gamma receptors blocked are pretty much overlapping with each other, meaning that there's not much contribution from Fc and Fc gamma receptor interaction. Then, we look into the binding kinetics between the CMG2 Fc to protective antigen by biolayer interferometry. We did experiment at 25 degrees and 37 degrees, and we found that under the same temperature condition, there's pretty much no difference in either the Ka or Kd value of all three variants, meaning that the binding kinetics between CMG2Fc and Pa are independent from glycosylation. This left us with the last possibility that the protein thermal stability could be different among those variants. And to examine this possibility, I incubate my three variants under identical at 37 degrees for 1 to 3 hours and 20 hours. And at the end of incubation, I did a functional ELISA to quantify the amount of functional protein that still remain active in the sample. And the result is shown on this slide. For the APOL and A-GLY variants, those curves are pretty much overlapping with each other, meaning there is no difference in the percentage of functional CMG2Fc during the whole incubation process. As for the ER-retained variants, there is a significant drop for the overnight incubated sample, suggesting the protein lost its function when it is exposed at 37 for a prolonged time. So this explains why we see a higher EC50 in the toxin neutralization assay, because we use 37 degrees for the toxin neutralization assay, which include over 9 hours of incubation. Then we look even further into this phenomenon by conducting molecular dynamic simulation. We first look into the secondary structure stability of all three variants, which I'm not showing on the slide. Basically, they all turn out to be the same. So the higher EC50 and the instability of CMG2Fc ER variants is not resulted from the instability of the protein folding. Instead, it's likely resulted from something else. Then we look into the final structure of CMG2Fc at the end of simulation as shown on the left bottom. The ER variants gives the most extended structure with its linker fully exposed to solvent. Then we look into the hydrophobic solvent-exposed surface area or hydrophobic SASA. The ER variants gives the highest hydrophobic SASA, indicating a higher potential for aggregation to minimizing those hydrophobic area. This higher hydrophobic SASA for ER-retained variants is also inconsistent with our non-reducing SS page, where you see additional bands above the full-length CMG2Fc, which is most likely representing protein aggregates. So from the molecular dynamic simulation, we see that the ER-retained variants gives the most extended structure and with the highest hydrophobic SASA. And that goes to the end of my presentation. Summarizing everything we have seen in the study are the following. First, we found glycosylation has stabilizing effect on protein during implanted production, evidenced by a higher expression level for the glycosylated variants compared to the non-glycosylated variants. Secondly, we found that the binding kinetics between CMG2Fc and protective antigen is independent from the unglycosylation profile. Third is that we found a higher EC50 of ER variants compared to among all three variants. And this higher EC50 is likely due to the loss of function at 37 degrees incubation during the toxin neutralization assay. And in molecular dynamic simulation, we found that the ER-retained variants, the higher EC50 of ER variants is likely resulting from the higher hydrophobic SASA, which indicating the protein has a higher potential for aggregation. So as for future work, it would be worthwhile to do in vitro unglycosylation remodeling to result more uniform glycosylation profile, allowing a better comparison between experimental and simulation results. And also, it's worthwhile to sequence the aggregate band to see what exactly is in there, whether it's intact protein aggregates or it's fragment aggregates, or it could be a mix of both. I want to thank Dr. McDonough and Dr. Nandi, as well as my research group for all the help, and also DTRA for providing the funding for this study, and DEB for giving me the chance to present my work here, as well as iBio for giving me help for toxin neutralization assay. Thank you.